our show. The discussions today, we have a lot. So, the first part of the discussion today is the immune escape and the possibility of that with Omicron. So, let's start our discussion. I'm going to start sharing and we'll go from there. So, first thing before Omicron's immune escape, this is Bloomberg. Toddlers make up 10% of hospital cases in Omicron epicenter. Now, I want to balance that with this. This is Mayo Clinic, and I would explain this in a second as well. How likely is it for a child to become sick with coronavirus? According to American Academy of Pediatrics and Children's Hospital Association, in the US, children represent about 16%. Of course, that news is about South Africa. Here we are looking at US. In the US, children represent about 16% of all COVID 19 cases. COVID 19 in children has been on the rise in the US. Again, this is children, not toddlers. Toddlers are 2 years of age. With children recently making up 24% of just over 100,000 weekly reported cases. Now, if I continue here, babies, so that is under 1 year. Again, they are toddlers, 2 years, up to 2 years. This is under 1 year. Babies under age 1 might be at higher risk of severe illness with COVID 19 than older children. This is likely due to the immature immune system and smaller airways, which make them more likely to develop breathing issues with respiratory virus infection. So, basic, and this is a true uh, immunology mechanism for all babies. This is why babies are given injections and protections, right? Some at the birth time. The reason for that is that when a baby is born, they still have their immune system cells being produced, their lymph nodes being produced, and their whole system being structured. And then the baby's immune system has to be exposed to antigens as well and learn to fight with them too. So they need some time to start developing their immunity, and till that time, they are vulnerable. Most of the time when the baby is born, they are given antibodies from mother as a gift through placenta before birth, and babies carry those antibodies with them for some time, and that provides some help. Then babies also get IgA from mother through breast milk if she is feeding them breast milk. By that time, we are expecting that baby would start to develop their own immunity as well. This is why, in the previous studies as well, we have seen that children 1 year and above, 1 year to 9 year, if they don't have comorbidities, they normally do not have COVID 19 related deaths. 9 year and above, they start having COVID 19 caused deaths, even if they do not have the comorbidities, but that percentage is really, really low. Under 1 year of age, babies actually have more issues because of COVID 19 because their immune system is not fully developed. So, now with this context in mind, if I go back here, South African scientists were last week the first to identify the new variant known as Omicron, and symptoms have been described as mild. The exact risk for the new strain is still uncertain. That is one. The very young children have an immature immune system and they are also not vaccinated, so they are more at risk. Then she said that so this is a doctor, she said that part of the increased rate of admission may reflect extra precaution on the part of the parents given the new concern about the mutations. So I think there is a part that may be related to the mutation itself. Then there is a part that toddlers actually have immature immune system. Then this may be that I think vaccine is a debatable discussion. And then parents may be a little more nervous too. So any sign of common cold or any sign of baby becoming sick, they must be bringing them to the hospital as well. So just wanted to make sure that we can look at it in the context. All right, so now for the discussion today. This is a table of various amino acids and their letters. Amino acids are the building blocks, the bricks that make up, that join together to make up our proteins. This discussion that we are going to do today is a little deeper in the mechanisms. So, if you're not comfortable with the mechanisms or how these structures effect, then this may be a, a discussion to skip. We will talk about more discussions after this one as well, but this is more of a deeper dive to try to understand how Omicron may behave. I also want to give you this context or this disclaimer up front that we actually do not have sufficient data. And so I saw some people commenting that if we don't have sufficient data, then why are we talking? And then there are some folks saying, when we are talking, why are we saying certain things that we don't have the data? And that is correct. But this is how science works, that as things start evolving, we start looking at them, we start sharing, discussing, and then from there we go on and we keep changing as more information becomes available. Cool beans who have been here for a long time, they are used to seeing these mechanisms, they are well versed with those mechanisms as well. So I think we can grasp this slightly gray shade of this discussion because this is more mechanism based, more theoretical, and then as more data comes together, we'll see more. Okay, so please keep this table in mind, especially this green area. And I want to give credit to this one part of the discussion to the person who brought it to my attention as well. So Stephen D. And this discussion was actually sent by him to copying me as well. And he was communicating with the authors of a few research papers that I would show you. So keep this part in your mind. Now we're going to go here. So the first thing, short answer. So if you just wanted to hear that, is the immune escape imminent? Short answer with the so far data and the behavior, no, it does not seem to be imminent. That is a short answer. There is lots of caveats and further explanations, which I'm going to go through. So here are the topics in which I will discuss this. Today's discussion is more of a discussion than the drawings. So this is a theoretical and mechanism-based deep dive into the immune escape possibility. So immune escape will depend upon many things, but some of these are amount of change for SARS-CoV-3. I'm saying 3 deliberately. If we are saying that immune escape will occur, then that would mean that we are saying that SARS-CoV-3 is coming together. If SARS-CoV-3 has come together, then we should look at SARS-CoV-1, MERS-CoV, and then see how SARS-CoV-2 was different from them and try to extrapolate. This is why I said this is a theoretical discussion as well. So if it is going to be a SARS-CoV-3, or let's say if Omicron is the stepping stone for the SARS-CoV-3, then we should see what kind of changes are there and are these sufficient to move this virus in a direction of SARS-CoV-3, or maybe these are sufficient to move the virus towards more humanized coronavirus. Then the type of changes needed. We have done this discussion in the past as well. A daughter virus may actually have changes that make it less stable, less proficient, less efficient, and as a result, it is not able to propagate too much. Or it could make it a daughter that is very similar to the parent, or it could make a daughter that is more efficient, more lethal, more transmissible. Various properties could appear. So question is, what kind of changes are we observing? So there are a couple of changes that I want to discuss. And again, credit to Stephen D as well. Epidemiological evidence. What are we seeing from the data in the society? The spread of it, how fast it is spreading, how much it is spreading, is it dominating as we're seeing from Delta and so on. Then clinical evidence. We will have to see that how does it behave clinically. If it is going to be immune escaping, then that would mean it would be a SARS-CoV-3 and a new pandemic will start. And that new pandemic will not care for who was previously infected and recovered or vaccinated and recovered. Everyone will become equal. So, if that is the case, we'll have to see that from clinical evidence as well. Then, of course, there will be researches, explorations that would happen. Then, of course, there is a question that how about the vaccine efficacy now? And what about the previous infection and the protection from there? So, I'm going to start from here. And we're going to go about these points one by one. First one, R naught or reproduction number. Reproduction number is the number that is used in epidemiology to say that an infection or a pathogen that goes from one person to one person, if one person sick with the pathogen makes another person sick, just one more person sick, so one person to one person, then we call it R naught of 1, and we call the disease to be stable in the society or community. If R naught is more than 1, that means one person is infecting more than one people, then R naught is greater than 1. And if R naught is greater than 1, then that will mean the disease is spreading in the community. If R naught is lesser than 1, that is, from one person, it doesn't even infect another one person, but lesser than one people, then R naught is lesser than 1 and disease is on the way out. It is reducing. Now, with this one, so far, and I use the word so far deliberately because this would change as the virus spreads, as the Omicron spreads, we will then see how it picks up momentum and velocity. So far, the data that we are seeing, preliminary data, initial data, says that R naught is 2. Compare that to delta's R naught, which was 3 to 8. It was from 3 to 8. That means one person with delta was making from 3 to 8 people infected. Here we are saying so far 2. So if you look at R naught, and if we take that on the face value, and we say this is it, we freeze ourselves in time with this R naught, then it is actually less spreading than delta. However, as more people become infected, and as they start spreading it to others, the actual R naught would start emerging. So keep an eye on the R naught in the coming days. So Google very often Omicron reproduction number. And if that starts going above 2, then that is a sign that the virus is becoming more transmissible, right? So that is one. Then, amount of change for the SARS-CoV-3. This is a very important topic, and I wanted to share this with you. So let's look at SARS-CoV-2 compared to SARS-CoV-1 or SARS-CoV and MERS-CoV. So if you see here, this is a discussion of SARS-CoV-2. But in this discussion, they compare it to the other cousins, other coronaviruses. So if you see here, SARS-CoV-2 was differed more from SARS-CoV at about 79%. So if I read this whole sentence here, the earlier Wuhan isolates also related with 88% identity to two bat-derived severe acute respiratory syndrome, SARS-like coronaviruses, collected in 2018 in Zhoushan, China, but differed more from SARS-CoV at about 79% and MERS-CoV at about 50%. So to become SARS-CoV-2, this coronavirus had to become 50% to 79% different from these previous cousins. What does that mean? What that means is, please keep in mind that SARS-CoV-2 has, or most of the coronaviruses, they have about 30,000, 29,000 some. So I'm going to just say 30,000 kilo bases, or the amino acids, or the genetic materials, or the bricks are about 30,000, 29,000 some. That is a very large virus. Because of that, this virus can become unstable very easily, but it has a proof reader that keeps it under control. Now, this 30,000 bases, if they were 50% different between the two viruses, so let's say this is MERS, and this is the genetic material of MERS, and this is SARS-CoV-2, SARS-CoV-2, then between them, there was 50% genetic material that is different. I just made it in one place, but of course this would be spread throughout the genetic material, scattered throughout. These changes will be scattered. Now, would it be interesting to see that the Omicron's changes are what percentage compared to SARS-CoV-2? So if we do that, Omicron has a total, so this is SARS-CoV-2. Omicron has a total of 50 base pairs changed compared to the parent. Now, many people have asked me that is this a set of changes on top of Delta? So no, that is not the case. This is not on top of Delta. That is, it has some changes that are similar to Delta, but it doesn't have all the Delta changes plus more. It has its own unique changes that have some Delta changes too and some not. So it's not on top of Delta. Meaning we don't take 50 plus we don't add another 18 from Delta or whatever number Delta had. So 50 base pair changes. Now 50 base pair changes means 50 divided by 30,000 multiplied with 100 is the rate, right? So that is actually, I think, somewhere like 0.1% or something like that. So hey Alexa, what is 5,000 divided by 30,000?
5,000 divided by 30,000 is approximately 0.1667.
So 0.16. 0.16%. So that means the percent change is really tiny. So then we are sitting here, we are scientists, we are looking at it together, and we're thinking this is a theoretical and mechanism discussion. And we could say, but maybe there could be a change in here, which alone can be drastic enough to cause an immune escape. And that is the second thing we should look at. So, first point here, for the immune escape to occur, the previous examples were such that almost half or more, 3/4 of the genetic material was different between the cousins. Here now, we're going to say, okay, this change is really 0.1%, now not even 1%. Can there be some one change that would really make a drastic difference? And one change that has been talked about nowadays is about the transmissibility. And you would see this a lot in various discussions, tweets, articles, that the cleavage, furin cleavage site has mutations. So this is important. So I want to show you this and discuss what does this mean. So keep this in mind. Furin cleavage site has mutations. What does this mean before I go and show you the changes? So let's say this is a spike protein. Spike protein has an RBD in it, receptor binding domain. Then it has an S1 unit and it has an S2 unit. When this virus attaches to ACE2, then we know that TMPRSS2, so here let's say This is TMPRSS2, which is a transmembrane protein. And I have done this discussion a lot in the past, so that is why I'm kind of just keeping it in a summary. Let's say here the TMPRSS2 will pick up a scissor. It's a protease. It cuts proteins, and it cuts here. There is a site here. There is a small thread here. That thread, just like if you go to a store and buy clothes, and the clothes have tags on them, and you cut the tag to sort of activate the cloth. So similarly, this little thread is cut. When that thread is cut, then the S1 and S2 become separated. The S1 can stay connected here, while this S2 piece that becomes separated, I'm going to make it in a different place now. Let's say it is here. That S2 piece would then fuse. It has become primed. It has become available. It has become uncovered by removing S1 from it. Now this S2 piece will fuse with our cell membrane and pull the whole virus in. So the SARS-CoV-2 has learned in Delta variant, has learned to make this area of furin cleavage much more efficient to be cut compared to its Wuhan cousin. I shouldn't say cousin, ancestor, because if it was cousin, there would be a lot of difference between them. There is a difference in clinical and phenotypical outcomes, but not enough to say SARS-CoV-3. Similarly, we're going to go to the Omicron as well. But let me hold this thought for a second and look at this one. Delta, compared to Wuhan, has an efficiency here in the furin cleavage site. It has a mutation here which allows it to easily break, which allows the TMPRSS2 to more efficiently cut it. That means it becomes primed fast. That means then it fuses quickly or more frequently, and so on. Now, Omicron has three mutations in the furin cleavage site. So the question is, are these mutations going to make it capable of becoming even easier to prime or not? This is, once again, I would keep calling out Stephen D's message. So I want to show you something. Here is the, I hope you can see this, but this is the Omicron mutations. This is Wikipedia. These mutations are present in many other sites as well. They have put the picture here. This area is, this green part is spike protein. If I can probably click it and show it, yes. So this green part is the spike protein, about 3,800 base pairs out of 30,000 complete virus base pairs. In this area, here near this RBD, there is an area of furin cleavage. If I show you that compared to Delta, here is Delta. You can actually see the changes. Look at the changes on spike protein in Delta. 1, 2, 3, 4, 5, 6, 7. And look at the changes in, I need to go back on this one. In this one, these are all the changes on the spike protein. So a lot of changes. But if you actually looked at the comparison, if you did a comparison, you would not find all the changes that are present on Delta to be present on Omicron as well. However, once again, keep this thought in your mind too. And I'm going to go to a third area. I told you today we're going to do some deeper dives. Here, These are the authors who were on that discussion that Stephen Lee did. So credit to him and the authors here. Look at this. This paper is, if I go to the top here, super antigenic character of an insert unique to SARS-CoV-2 spike supported by skewed T cell receptor repertoire in patients with hyperinflammation. So their point of view is something different, that they're saying there is a part in the furin cleavage area which has mutated, which allows the virus to actually move towards hyperinflammation as well. But I want to use some other part here from their discussion. That is this 681. This is the 681 amino acid on the spike protein. So spike protein is 3800 amino acid long, or 3800 bricks long. The 681st brick is the area of furin cleavage site. So 4-5 amino acids over there make up the furin cleavage. And this 681, can you please look at this one? So this is PRRA. That is the genetic sequence. So if I go back here, PRRA, from here you can see what these things are. So R is an arginine, and then P is a proline, A is an alanine. So if I go back here, these are bricks names. Every brick has a different structure, just like when we build our houses, there is a brick that makes a corner, and there is a brick that can make a part of the roof and ceilings and so on, or chimneys. So these bricks are various structures and various electromagnetic forces, and they bind and give shapes to they bind together and give shapes to proteins. They make and shape the proteins. So if I go back here, PRRA, furin cleavage site, it has proline and then RR and then A. What was RR? I forgot the name of R, arginine. So proline, arginine, and alanine. Now let's go to delta for a second. In the delta, in the furin cleavage site, there was a change that's, if you see here, this change is P681R. So proline here at the furin cleavage site. And previously I was pointing somewhere over here. This is the area of the furin cleavage that is between S1 and S2. So P681R, what does this mean? Delta variant had a change that P proline was replaced with arginine. Now let's look at this guy. And if I go here to 681, do you see here? Can I move it? Check this one out. P61 with H. Sorry, so in the previous one, proline was replaced with arginine. Here, proline at 681 position is replaced with histidine. So if I go back here to the decoder, this area is histidine. Now I want to show you something here. The charge on arginine and histidine is same. So the furin cleavage site on delta and on Omicron, from a charge point of view, is basic. They both are basic. They both are positively charged. That means the electromagnetic forces over there may not have changed too much. The shape may have changed, but these shapes also may not be changed too much, because most of the time when there is a kink in the protein that is produced, usually that is because of the branching amino acids. So if you see here, there is, where is the, so if you look at glycine, for example, it has a branch in it. So there are amino acids with the branches that can bend the protein. These amino acids mostly are in the similar shape and similar charges. So that means the delta's furin cleavage site and the Omicron's furin cleavage site is actually very similar to each other, which means that, so if I go back here, it is possible that the TMPRSS2's behavior is unaffected. And if that is unaffected, you might see the behavior very similar to Delta. Yes, it is a new name, it is a new variant, it has a lot more mutations. Every mutation would have a behavioral outcome. Some would have no outcome. Some may have a positive outcome. The others may have a negative outcome for the virus. Positive, when negative for the virus, positive for us. When positive for the virus, negative for us. So here, it seems like furin changes may not be. I'm using again may deliberately. I'm not trying to avoid a statement. I just cannot make a statement because we actually do not know enough. So we're looking at mechanism. It may be that there is no change to the priming of it. And if there is no change to the priming of it, there may not be much change to the way the furin cleavage site works. That doesn't mean that the virus can still not bind more efficiently. There are 30 more changes which may make the virus bind efficiently with better electromagnetic forces, with better shape. But this priming of it may not have become more efficient. And one can go over each change one by one and kind of start extrapolating. Again, this is theoretical and mechanistic. What is the clinical outcome? What is the epidemiological outcome is not known yet. That data is coming together. So I wanted to share this one thing. Now I'm going to go from here to the second topic, which is really important. And that topic is, are we going to see antibodies not work anymore? Antibodies from the vaccine or antibodies from the previous infection. And maybe I would go with this statement so far that antibody production or antibody efficacy may have reduced. But even for that, we need data to see how much reduced. Just like, for example, with Pfizer and Moderna and Johnson & Johnson, as the time is passing, we are seeing that the efficacy generally is reducing. How much with this is that reduction in efficacy is accelerating? Don't know yet. That will have to come together. But the question is it possible, once again, that it just fails? The antibodies just fail to work, either from the previous infection or from the vaccine. So that is what I'm going to talk about now. So once again, I'm going to take this diagram and say we have SARS-CoV-2 about 30 kilo base pairs or 30,000 bricks. We also know that the spike protein, if I look at this, most of these references are in the description as well. So the spike protein is encoded by 3,831 base pairs of SARS-CoV-2 RNA. So 38, what is the exact number? 3,831 base pair long base pairs. So that is the structure. We have done this discussion in the past, that a part of the protein that can be exposed to an antibody to which the antibody can bind is called an epitope. Epitope. A piece, a pattern. Now, these 3,831 will make a protein that is three-dimensional protein, which we call spike protein. And you know that it has various pieces to it. And just very quickly, to show us the three dimension, and there is a point I want to make with that. If I go here, and this is a spread: Italy, Spain, Australia, Austria, Germany, Czech Republic, Hong Kong, South Africa, Belgium, Canada, and Botswana, UK, Reunion, Israel, and Netherlands. Anyways, back here, I want to look at the structure. This is a three-dimensional structure. You can actually think from this structure that it is not necessary that every single pattern of this protein will be available for antibodies to bind to. I think it is reasonable to understand that only those patterns that would emerge on the surface of this three-dimensional structure are going to be available for antibodies to bind. So that is, if you look at my hand for a second, let's say if I make this little fist here, there are patterns that are inside, and then there are patterns that are outside. So imagine if all of them together are 1000 patterns. But when I close my fist, 500 patterns have become unavailable because they are hidden, and only 500 patterns are available to be bound. So the first thing to keep in mind is that it is not necessary that all 3831 patterns will become available, or amino acids made patterns will be. These are not 3831 patterns, but these are the breaks. So then what is available? Whatever is the structure and things that are outside, these are the available one. And epitope usually is anywhere from 7 bricks, 7 amino acids, to 21, depending upon we're talking about T cell receptor or B cell receptor and so on. So let's assume 10, just for our discussion sake. Let's say 10 amino acids together, 10 bricks laid together will make one pattern that can be recognized and bound by an antibody. So then if we have 3831 base pairs, if we divide them, then that would make linearly, that can make 383 available sites. Now, some of them are going to get folded and go in. So what is available outside is less. That's one. Number two, the availability of a site is not linear. That is, let's say if I am writing A, B, C, D, E, F, G, H, I, J, K, L, M, N, O, P, Q. Now let's say 1, 2, 3, 4, 5, 6, 7. Let's say this is one pattern. It is not necessary that the next pattern has to start from H and has to go H, I, J, K, 1, 2, 3, 4, 5, 6, 7. This is not the case. One can actually have patterns that are from here to here as well. Let's say from here to here. So now within the 8 amino acids, there are 2 patterns. And then within the 9 amino acid, there could be one pattern here, the second pattern here, third pattern here. So there can be overlapping patterns. That makes it hundreds and hundreds of patterns available. What is the point of this? The point is, when the virus arrives in our body or the vaccine, it is not that there are just 2 or 3 antibodies formed and we are done. And then there are changes to the spike protein and we have a problem. In fact, there are going to be hundreds of antibodies that are going to be bound to various patterns, various epitopes. And they could even be overlapping patterns. Now let's say out of these, there are 30 changes, 30 bricks have changed. It is not necessary that every single change would fail a single relevant antibody. Some changes are silent, they don't even matter. The chemical and electromagnetic and phenotypical, or the shape and the electromagnetic forces stay the same. So even if there is a slight change in there, we say, well, this is a silent change, nothing happened. Some changes are drastic. When that change occurs, the whole thing just falls apart. This is equal to um, installing a door but taking the hinge of the door off. So that may be a small change in the whole construction, but taking the hinge out will drop the whole door, would not work. So there are some changes that could be bad, and then there are some changes that could be silent, and then there are some changes which are just average changes. So the point is that we have 30 changes here out of hundreds of epitopes. Now, these 30 changes are not necessary that they are making 30 epitopes. They could be making 10, or they could be making 50 because of overlaps. But in the bigger picture, there still are a lot of epitopes available. Now, if somebody had the previous infection, they would have epitopes not only for the spike protein, they would also have they will also have antibodies against their M protein, matrix protein, their envelope proteins, their HE proteins, their N protein. So there are much more set of proteins. There are 30,000 base pairs, right? So there are much more epitopes available for their antibodies to have attempted to bind with. For a spike protein vaccine, for example, those number of epitopes may be less, but again, sufficient to prevent a serious disease. So, in my opinion, so this is an opinion part. After presenting this mechanism, I think that you can make an opinion of your own. You can hear my opinion out as well. And then, as the data comes together, we'll see the reality is what. So, my opinion is these are not sufficient changes to cause immune escape. That is to say, all the antibodies that have been produced by a vaccine or a previous infection cannot now work on this virus. Actually, actually, I find two things very interesting about this, and this data is preliminary. One is R naught of 2, and the second is incubation period of 10, 11 days. 11 days long incubation period tells me that the virus is struggling to cause damage. Now, if this data itself is wrong, then the statement that I just said is wrong as well. So maybe I'm wrong. We'll find out as the data emerges more. That's not a matter of debating. It's not that somebody would come and debate with me and say, no, it is not 11 days, because you're wrong on incubation. We'll just know the data soon. But are we now becoming escaped? Is the virus becoming escaped from our immune system? I don't think so. Previous infection, even better protection, because they have antibodies against M and N and other parts. And vaccine protection as well, because there are antibodies. Now, the question of efficacy may be an interesting one, right? And that is, it is possible that a vaccine has generated some antibodies, and then let's take a number for our purposes. It may be a hypothetical number. Let's say the vaccine has created 100 antibodies that can attack the spike protein. Out of those 100, there are 10 that have become disabled. They cannot bind correctly anymore because of the shape changes. If that is the case, we are now left with 90. Are they sufficient or not? Or maybe we are left with 20 and 80 have become disabled. We don't know that yet. So that is the piece of information that would come together either from research, where researchers will pick up the spike protein and try to bind it with the antibodies from convalescent plasma, or antibodies generated from the vaccines or monoclonal antibodies and see what is the binding affinity and what is the efficacy. Then we would also see that from clinical outcomes to see that people who were previously vaccinated, for example, that outpatient situation which Dr. Angelique Kotzia said, she said that I have more youngsters that have the infection milder. Her words are extremely mild, but she also said 50% vaccinated and 50% unvaccinated. So if that is the case, is that different? Was she previously used to seeing, let's say, 10% vaccinated and 90% unvaccinated? And is that changed? That is what we don't know yet. I'm actually trying to see if I can have her on our show. But that clinical evidence would start telling us what's happening. The second thing, let's say reinfections occur more. The second question to keep in mind and the observation to do is going to be, what is the disease progression? Is the reinfection milder, for example, common cold occurred again, or it is bad? And then reinfection is really causing lots of problems. Then we would start thinking about, at least I would start thinking about that, hey, this is now going from SARS-CoV-2 to SARS-CoV-3 or maybe SARS-CoV 2.5. So these are the two important points that I wanted to make sure that we look at. So if I go back here to the questions, what is the amount of change? So this virus does have amount of change much more than Delta, but to make it a new cousin virus, make it SARS-CoV-3 and have the pandemic start all over again, that's not where we are, at least not from the data we are seeing and the changes we are seeing. Type of changes, I just discussed furin change, courtesy of Stephen D. Maybe every single change can be looked at. There are two ways to look at them, mechanistically, just like we did, or clinically, or together, or with studies in labs. Epidemiological evidence, at least from the news, it seems like it is spreading fast, it is becoming dominant. That means if Delta is standing here and Omicron is standing here, and they are both asked to open the door and get into the cell, Omicron seems to be more efficiently able to do it. That means we might start seeing Delta's name fade away and Omicron's name become more uh, in the news. Then clinical evidence. This one is still up in the air. From Angelique Kotzia's point of view, milder cases in outpatient, they can be treated at home. But most of the outpatients generally can be treated at home. That is why they came to outpatient. If they had bigger problems, they would end up in a hospital. There is Dr. What is her name? There is another doctor from ICU. She said she's seeing more 20 to 30 years of age patients in ICU in more severe disease form. She said severe or critical. She also said something which was very interesting for me. She said 65% were unvaccinated and the rest were half vaccinated. I find that difficult to believe. I'm not saying that it should be, she's not saying correctly, but there would be some vaccinated as well. It, I don't know what percentage, maybe a smaller percentage. But, anyways, this is her message that 20 to 30 years of age are more ending up now in ICUs. They have more severe form of the disease compared to previously, and they are mostly unvaccinated or half vaccinated. So I think that we have to keep our mind open to learn more in that area from more doctors, and then researches and trials would happen as well. So clinical evidence is emerging. Research and exploration, I'm sure that it has started. If you look at WHO, they've said that, hey, we have started a lot of research as well. And then these are the discussions that we have already done. So this is the first part of the discussion. I'm going to stop here. It's almost 50 minutes. And I will then come back and we will do one more talk about how various news articles are seeing things. Then we'll come back for a chit chat. And if I could muster up the courage today, we'll do a chit chat with the questions only from YouTube. Then, after that, you can go to sleep, and I'll do one more chit chat with the questions from Twitter. And tomorrow, somewhere during the day, I will try to do one more chit chat with the questions from Patreons. So that is the plan. I'm going to see you in a few minutes. And please do me a favor, like, subscribe, and share. And if you would like to support this work, there are links in the description. You can buy me a coffee, or you can become a patron, or you can use PayPal to support this work. Thank you very much. See you shortly.